Welcome to New York Bio's Virtual Breakfast Series, a digital program started in 2020 bringing you fireside chats with leaders across the healthcare spectrum. This week's episode features Dr. Neal N. Padte, Founding Chief Operating Officer and Chief Development Officer of RenBio.
Good morning, everyone. We'll get started. My name is Jennifer Hawks-Bland, and I'm the CEO at NewYorkBIO. We are very excited you're with us for another edition of our virtual breakfast series today. A special thank you to our sponsors for this month's series of breakfast, Iser Amper. We are going to talk to Neal about all kinds of things, startup, founders, tech transfer. We are going to cover the waterfront and starting a business in New York, our favorite topic of all. As far as housekeeping, as Derek and I go through the conversation with Neal today, we know you're going to have questions, so please ask those in either the chat or the Q&A boxes on your screen, and we will get to them as we can throughout the course of our conversation. With that, I will turn it over to Derek to kick off our conversation. Thanks.
All right, thanks so much, Jennifer, and good morning, Neal. It is wonderful to have you here. Post Thanksgiving, we have a leftover plate of everything. We have founder-led biotech. We have people coming out of academia in New York. We have a bit of tech transfer. We have cool new platforms. We have great science. We have everything. So it is wonderful to have you here. You are an absolutely wonderful startup story out of New York City, and we're excited to dig in. So before we kind of get into the whole thing, why don't you give us a little bit of an origin story and tell us how you got where you are today?
Sure, happy to do so. And thanks, Jennifer and Derek, for having me. I'm really excited to chat with everyone today. I think when I think about my origin story, I realized I tend to like this balance of creativity and driving things forward. And I found now when I reflect on my career, I've kind of oscillated between the two in different forms. So I found a way to connect the two, which is where I am now. So by training, I have my PhD from Columbia University. I was in a basic cell biology lab. And by basic, I mean, we were studying cytokinesis and cell polarity in fission yeast. And I joined it because I loved the professor I got to work with, Fred Chang, who's at UCSF. I thought he was going to be a great mentor. What I really loved about that time is, you're working on genes that no one knows what they are yet. You're working on proteins, no one knows how they work, and you really don't have a big tool set. There were maybe 2 or 3 papers in the field on what we're working in, but it really taught me how to get comfortable in that black box and get comfortable pushing what I call, what was really exciting to me is that you live on the frontier of knowledge, and you're just trying to poke it a little bit at a time and see if you can just push what humans understand. And that part was really exciting. But from that creativity side, I realized I was missing the translational side, which is really where I wanted to end up and just see medicines get into people and move products forward. And I realized kind of midway through my PhD that wasn't going to happen in a basic research lab. I was lucky to be at Columbia University, where they were just starting at that time. This was the early 2000s, the Tech Ventures Fellows Program. So this is led by Oren Herskowitz, and Brenner Rao at the time was there, who's at Modern Meadow now. And this is an amazing program where they basically took PhD students who were kind of towards the end of their career, or studies, I guess is the right word, and had them evaluate the tons of inflow of different inventions and technologies being submitted by Columbia professors. So we'd be the triage, if you will. Every week you get a new technology. It could be cell biology, it could be a vaccine, it could be, back then it was touch screens that became part of the iPhone later on. And our job was to do a deep dive for a week and come out of it, see if that was patentable, do some comparison on what's out there. And that really it was like being a kid in a candy store. Every week you get a new topic and you get to learn new things. And it was really, really a lot of fun and showed me what's out there.
That's awesome.
Yeah.
Was it super competitive to be selected to work with?
I think so. It was the early days. It was very interesting. It was still secret back then. You weren't really supposed to talk about not becoming a professor. I was very lucky. I had a professor that I was very open with right around midway through when I realized I wasn't going to go into academics, and we talked about it. But so you kind of met your crew, your people as you went along. But it was just forming, and I think there's other universities that do that as a model now. I have a feeling Columbia was one of the first, which is really great.
Yeah, and it's interesting. It's actually a really good thing that you had a healthy enough relationship with your PhD mentor to have that conversation. I don't know that everybody is so lucky. I think the barriers to this are breaking down a little bit, but I think there still is, in certain circles, I don't want to call it a stigma, but there is basically this expectation that if you go through so-and-so's lab that you follow in the footsteps and you blaze your path into academia. Did you know other people that kind of were under pressures like that?
Yes, I think a lot of people there were. And when you find a group like the Tech Ventures Group and this fellows internship, you find other people that are trying to look for their way out. I think I was very lucky. I didn't realize that I was essentially doing annual reviews with my mentor, which wasn't a thing in grad school. But every year I would sit down and say, hey, can we meet? I want to review what I did this year, what I think was good, what I think could be better, and what my goals are for the next year. And looking back, that was probably the best thing I could have done. It was initiated because that's not exactly how professors are trained to work with their students, but it just gave me room to talk to them on a very regular level. And as it started to evolve, we were able to talk about it. So I think I was pretty lucky in that regard.
Yeah, no, that's both luck. Well, that's both lucky and smart, right? So, you were lucky you had the foresight to do that. So out of your PhD, you landed in what I imagine is this just really cool, because if you wanted to do translational stuff, you could not have landed in a better place than the Aaron Diamond Center at the time that you entered. I'm gonna tee you up with that, but it seems like you landed in kind of like the ultimate place for translational science.
Yeah, that's a great tee up because it's true, and I loved it. So I went right from grad school after getting my PhD to the Aaron Diamond AIDS Research Center. At the time, it was the largest HIV/AIDS research center in the world. It was a nonprofit affiliate of Rockefeller University at the time, but now it's at Columbia. They do everything from basic cell biology of HIV all the way to clinical trials. And it's led by David Ho. So some folks know him very well. Some are still learning who he is. He was Times Man of the Year in the mid-90s, Presidential Citizens Medal from Bill Clinton for essentially laying the groundwork for triple combination therapy that changed HIV from a death sentence to a livable disease.
I was going to say, I think it's fair to say that he is one of the absolute pillars of science, and one of the main reasons that HIV is in a state it is today in terms of actual therapeutic options. I think there's no there's no way to cut around it. He is a huge figure in terms of impacting that landscape.
Yeah, I would completely agree. And I got to work with him, which when I had the opportunity, I said, look, my goal is always to work with smart people and learn from great people out there. And when I had the opportunity to do that, I took it.
Okay. So what did you do for them?
So I was hired as a scientific project manager, which I had no idea what that was when I took the job because I didn't know what project managers did. They were looking for someone that had a PhD level background to work with the postdocs in the lab, and they were just funded. They worked, with small molecules and treatment for many years, and they were just moving into antibody therapeutics for HIV prevention and treatment. And that was very new at that time. That was still just getting talked about. They got a large grant from the Bill and Melinda Gates Foundation, and Bill and Melinda Gates Foundation was really interesting because I think they empowered these academic groups to do drug development for major public health challenges that may not have necessarily had huge commercial markets, but needed to be solved. And so HIV prevention throughout the world, the developing world, is one of those. Gates Foundation was smart because they required these labs to have a dedicated project manager. And most of the academic labs didn't want to do that because they didn't really know what their purpose was either. But they brought me in. And so it was interesting. I said to myself, okay, this would be a great way just to learn about all the different pieces of drug development, how it works. See the discovery side of antibody development and see it all the way to the clinic. And I told myself I'll stay for a minimum 2 years just to see which aspect of drug development I liked. And then I figured I would just pick one and go down that path. I hadn't exactly done that because I realized I liked all of them and I wanted to stay there. What I told myself when I started was I will stay as long as I can keep on learning and growing. And Aaron Diamond was one of those places that was really just fertile ground to really grow. It felt like a nonprofit biotech in that translational research group that I really, really loved. So I was hired when they took their first program. It was at Discovery. It was the first bispecific antibody against HIV. It was about 1,000-fold more potent than anything that was discovered before. The Gates Foundation wanted to take it from discovery all the way to the clinic because that could have impact throughout the developing world. If it's 1,000-fold more potent, you can give significantly less levels. There's a lot of other things in the target product profile that are great. So David Ho was fantastic, and the organization was great because essentially, like a biotech, if you need to do something, if something needs to be done, if you volunteer to do it, they'll give you the opportunity to try it if there's no one else there to do it. So when we got to this stage, we did discovery, early translational work, and all of our clinical work was always with industry partners. This is the first time we were taking a program completely ourselves. So doing the CMC manufacture, supervising the GLP tox, submitting a commercial IND package in the trial. So I just volunteered. I said, hey, I can start this process because honestly, as a project manager, I just need to get my programs to move, right? I had my own kind of internal metrics I wanted to hit. So I started sourcing GMP contractors. I got to supervise the manufacturing campaign. I started reviewing every single batch record, signing off on them. Did the same for the GLP tox program, did the design. The commercial IND package, there were 4 of us that wrote the whole thing, which was amazing. Like I remember talking to David and saying, don't we need to hire a few people for this? I don't know if we could do this. And he said, Neal, you'd be surprised what you can accomplish just with a little bit of insight and thought and taking some time. You don't want to get too stuck in the ways that it has to be done and lose that creativity. Just be thoughtful, know when to ask the right questions, know when to seek guidance from others in the field. And you'll be surprised what you can do. And so I said, okay, I guess we're going to do this. And the 4 of us wrote this. We got it through to the FDA. They gave us a call, said everything looks great. They gave us a fast track approval to take it to the clinic. And then it went into phase 1. And I realized at that point in my career, I really loved not just creativity or getting things done, but that connectivity. And seeing all the pieces was super exciting. And to go from seeing data that was on an Excel sheet to become a medicine that caused a physiological change in someone was something that was just awe-inspiring for me. Like I had no idea I'd ever get that possibility to be in that environment. And I knew I wanted to find ways to do more of it.
And all of this, Neal, was in what time frame? How long?
So we identified our lead to the clinic was 2 years, 2.5 years. I think. So this was around mid 2014 to 2016 in that range. Yeah.
So was there ever a point in that process where you just got hit with the wave that you weren't expecting? Right. I mean, there's a whole level, whether it's with this or whether it's with a biotech company or anything like that, where when you do something where you don't know what you don't know, where you kind of find out why people say it's hard. Right. You're like, oh, yes, this is why people, treat this like a big deal, because it actually is. Was there a point in that process where you kind of got hit with all of that at once and were kind of like, yeah, okay.
So there were, I'd put it in 2 categories. I think specifically what you're saying, that happened a lot. And then I just became not shy about asking questions. We were still in an academic group. And so I just tried to lean in on that where it could help. You try to read some literature, like even toxicology designs. We had a unique molecule. It's out there how to do this. We had some consultants helping us, but we were doing it in a unique version. I found some papers written by some industry folks that might be relevant. So rather than me, we had a postdoc in the lab working with us. I said, hey, can you email this person? Their corresponding address is there, and just see if they're willing to talk to us. And the scientific community I was really happy to see that they offered their knowledge to us, right? Because we were this academic group trying to make an anti-HIV preventative therapeutic. And we just picked up the phone. We talked to some folks at Roche, some folks at Biogen, and they just gave us, only publicly available information, but a lot of guidance. So
yeah, but publicly available is fine, but actually knowing where to find it, knowing what it is, knowing what's hard, knowing what's good and knowing what isn't, that's kind of that's kind of the big value there. That's good to hear.
So you just have to be resourceful, I think, and not give up. I think people have said it before, tenacity is such a big part of this, and just knowing that there can be a path. I think even my basic research training from grad school helps. You sit there without knowing the answer and you're like, all right, we're gonna figure this out. The other side of when you said, you get stuck, I feel like every drug development program I've been in, there's 3 distinct moments where you think the whole thing is going to crash and die. And you have a night, you go home, you have a drink, you're like, all right, we're going to figure this out. And then
let's see if this looks the same in the morning, shall we?
And you get a surprising result during your manufacturing, or it's not as stable as you thought, or like it crashes out of solution, or you think you might see a safety signal in early animal studies. And it's an interesting, everyone thinks it's kind of, when from the academic side, they said, oh, that's the easy part, the development. The hard part is the discovery. That part, it's already wrote, but there are so many places where it can go off the rails, and you've got to just bring it back and drive it forward, or it'll never get done. So that's something I've kind of learned, that tenacity goes with that as well. You will have a moment where you think everything will die. And you have to be honest, sometimes it might, but most of the time there's a solution.
Where you sit is usually the most difficult part, right?
Right.
So the
Right. If you
academics think theirs is, yeah.
Of course.
You,
Yeah.
it's literally the describing the elephant piece, right? If you talk to anybody in
Yeah.
biotech, the piece
Yes.
that they work on is the hardest and most important piece of the whole. There are very few people that you meet in the industry be like, actually, what we do is kind of easy. These guys over here
Can
in
you
discovery,
imagine?
that's actually the really hard part. Nobody wants to do. That's where all the real smart people are.
It's true.
So how did it work? I mean, so you guys took things you guys took them through phase one, right? So how did it
Correct.
work? Did you have a plan for what was going to happen later in development? Did you have kind of either a BD or tech transfer function in there? Like what was the plan for phase two and phase three?
Yeah.
Or was Gates ready to fund phase two and phase three? Or was it all kind of, let's see how we do in phase one first? Because this was kind of the first foray, right?
Right. So back then it was, let's see, it was a lot of translational research. It was experimental translational medicine,
Yeah.
experimental clinical medicine. Let's see how it looks in phase one. A lot of those programs in the early day, you were getting that phase one data and you're going back and you're making either new vaccines or new molecules and bringing it forward. Because in HIV, there's things you can only learn in humans because the animal models just aren't designed. They're not as relevant for all the questions you want to ask. But for this program, Gates Foundation was the first one that pushed us because when we're applying, even to take it from preclinical into the clinic, they want to start talking about, all right, part of the application is how are you going to make this, they call it globally accessible. So
Right.
it wasn't commercialization, but it's how are you going to get this to phase two?
Right.
Who are the partners? What is phase three going to look like at a high level?
Yeah.
So we started thinking about it. And when it gets in the clinic, we started talking with the foundation about it, what we'll do to go forward. We're actually partnering that molecule now, Iron Diamond is separately with the US HIV military research program. They actually have a huge arm that's doing work that will take on a phase two study as well. So that's the goal right now, to take that forward.
Yeah, I imagine at least within Gates and kind of the pioneers on that one were the Milken Institute, right? And actually the Prostate Cancer Foundation. I think he was the first person really to say, if there's no commercial path for this, we're going to make one because this is important to patients. And I think other foundations, Gates in particular, have been smart enough to say, we're not doing this just to discover a cool molecule and hope someone takes it. Like we need to have a plan to actually get it to people, which is really the biggest thing. And it's funny, and hearing you talk about all of this stuff now, right? It is really kind of the perfect minting ground and training ground for eventually leading a biotech company, right? You've dug in and you're saying things like, well, the animal models weren't all that great. They don't really translate to you're kind of ticking off all of the things that end up being these problems that you have to figure out when you start a biotech company or you blaze into a new area.
Yeah, totally agree. It was great training ground. I think having David Ho as a mentor was amazing through this process. Speaking of that, as we were starting the company, it was interesting. So every presentation. So as I progressed at Iron Diamond, it was wonderful. They were as I got promoted, they were creating positions for me at each level that never existed at the company because they realized I was doing it. So they're like, let's find something that reflects what you're doing. So by the time I left, I was director of product development. And I was giving presentations at conferences, at meetings with the Gates Foundation about our work. Every presentation I ever gave, usually with the academic groups, there was always a question saying, this is great, fantastic, really promising what you're doing, but come on, antibodies are really expensive. How are you going to think this is actually practical and realistic for the developing world to make?
Yeah.
And they weren't wrong, right?
It's
a challenging question.
It's a legitimate question.
And
I
my
believe the
answer
answer
was
always started with, that's an excellent question.
right. Take a
point.
second. But I was thinking about this and our kind of our ethos behind this, and you're right. It's not practical long term, but the reality is nothing exists today. You've got to start somewhere. You've got to get something to help these people that are dying. And if you can show impact, the innovation will come around it, right? Look at where we are with the iPhone, right? And how big computers were. When people saw the promise of what that was, then you get all the engineers that come in, all these other folks that come in that make it more practical. So we would say, look, nothing exists. We got to get something to at least stop this disease. And then if that happens and it's promising, the innovation will follow behind it to make it even more accessible. So it's a really interesting place. And I think biotech too, because you have to be optimistic, but as scientists, we're trained to be very cynical, right? So how do you balance the two? You've got to poke holes in everything, but you've got to have some hope that this is going to work. Otherwise, you can always talk yourself out of it.
Right. So I want to hear the origin story for where Ren Bio came from, because I will tell you there there are a few of these. It doesn't happen very often, but when you and I first met and talked about this, and you told me about what NewYorkBIOs platform was, my immediate reaction was, oh, that's really cool. Right. There are certain things that are kind of like twists and plays, and you go into this area and stuff like that, but there aren't that many things that, kind of flip a switch and be like, oh man, if that worked, that'd be really awesome. All right. So, with that as set up, tell us a little bit about where this came from, and you can give us your TED Talk pitch about how awesome NewYorkBIO is, because I think this platform is fantastic.
Cool. Yeah. So it actually leads from what I was just saying. So I was giving these presentations, right? It's not practical long term. And we said innovation will come behind it. So we actually ended up working on that next generation approach, which became the formation of what NewYorkBIO is today. So while we were taking this antibody forward, we said, all right, antibodies are expensive to manufacture. HIV field was doing DNA vaccines at the time, so which were a little more practical. They could be thermostable, temperature in sub-Saharan Africa. They can last much longer, easier to make. So it was a very simple question. We said, look, if all the information for antibodies are in the DNA sequence, do you really have to go through these larger and larger bioreactors and purify it and then infuse it? Can you simply just give the DNA that encodes for that antibody intramuscularly and have your muscle cells serve as your in vivo bioreactor that produces your antibody for you? Then you get around all the challenges of making it and even the infusion process, because you have the instructions on board. It's not permanent, and that's by design, but it's much, much longer than a typical antibody.
Right.
And we said, if this could work, we started with saying, could this work for HIV? And I still remember the day. It was me, it was David Ho, and Yaoxin Huang, who's the other co-founder of NewYorkBIO. These conversations always seem to happen like on holiday weekends. Like it's the Wednesday night before Thanksgiving. You very quickly realize you're going to text your family that you're going to be home late. This isn't going to work out. Could you just get in this conversation when everything gets quiet? And we sat around and David said one day, look, we're working really hard to make this work for HIV because we have such a high bar to meet. Our target product profile of, thermostable drug product. We can't have infusions every couple of weeks because you can't expect someone to walk 5 miles from the village to the local clinic to get an infusion. They're not going to do that. So we're working on this DNA encoded approach, and we still don't know the right antibody for HIV. It's early stage. There's a whole industry and field of antibodies we know work for oncology, for rheumatoid arthritis, that no one's innovating in this way because they don't have that same bar or that same challenge of ours, which was making this accessible for the whole world. So if we took that idea and now moved DNA encoded antibodies or therapeutic proteins into these commercial indications like oncology, rheumatoid arthritis, that could be a game changer. Now you're not sitting in an IV infusion clinic for 3 hours. You're getting a 30-second intramuscular injection and you're out the door. You're not coming in every 3 weeks. Now you're coming in once every 6 months. And the goal was at the time, come in once a year for your antibody shot, just like you would get a flu vaccine or something like that. And it's thermostable and easier to make. So it could work for the developing world and bring access to these antibodies to people that have never seen them before, but also really improve antibodies and proteins in the developed world as well, where no one was thinking in this way. So what I didn't mention at ADARC is, Aaron Diamond, when I was doing all the drug development work, in drug development, you've got a million CDAs, MTAs, clinical trial agreements, all these things. And we had administrative office that did that, but they had 6 other labs they were working with all these different NIH grants. And I need to move this program forward. So somewhere along the way, I just volunteered with the COO that was there. I said, hey.
I sense this theme.
Yeah, really.
It just kind of happens. I said, can I just like pre-review this for you? Pull out the salient points. Let's have a 15-minute meeting and just move this forward because I needed to get this forward.
Yeah.
I've learned now in another position, no one really rejects that offer. I didn't quite realize. I was like, is it okay if I can like help out? She's like, yes,
do the work.
Sure.
Yes.
So I would just start pre-reviewing and I would just learn from her because I wanted to know how she did this. And she had a lot of experience in this. And gradually, I just started leading more of those legal negotiations. We out-licensed a couple molecules. I got to be part of the deal teams that did that work. I started overseeing the multi-million-dollar budget to do this. So when we started NewYorkBIO, David said, Neal, by this point, you've already seen a molecule and a drug go from discovery to the clinic, and you're doing a lot of the legal negotiations, finance work. Why don't you go spin out NewYorkBIO and make this thing happen? And I said, absolutely. I always wanted to get into the industry side of things. And I said, let's do it. And so that was NewYorkBIO. So we took this idea of a DNA encoded approach. So the goal here is to basically make antibody therapeutics more durable, more efficacious, more cost-effective, and more long-lasting for people,
Mm-hmm.
and help people in the developed world, but also get these really powerful antibody and protein therapeutics to the developing world as well. So get it to be globally accessible. And so we did that. So this was before, the idea was super high risk at the time. There was not a lot of data to support this would work. So there weren't VCs coming in those early days to see this happen. And so we ended up just pitching and getting money from a group of angel investors and high net worth individuals that had a bit of a philanthropic bent as well, and wanted to see this move forward, has seen what David has done previously with his other innovations and programs, and said, sure, let's see what you guys have. And so we raised a $4 million Series Seed from them. And then I went out to start the company. And that kind of started the same kind of theme that I was doing at ADARC in drug development. There was no group to really teach me how to negotiate real estate leases, develop HR policies, hire a team. And it's a theme when you learn from David Ho. You figure it out as you go along and just know when to ask the right questions. I thankfully had some other mentors I can rely on or people I ask questions to, but you just figure it out. And I don't think that story is unique to me. I think a lot of founders do that, right? You figure it out and you do everything. And the company was born. So that was in 2016 when we opened our laboratory doors at RenBio.
You also had the benefit of working with DARPA for a while too, right?
Yes, that was huge. So we had these angel investors come in to seed us, which was great. And then DARPA is really great about taking high risk projects and seeing the potential. Back then in 2016, they said they want to develop technologies to prevent the next pandemic infectious disease outbreak. And they saw our technology as a tremendous way to do that. So Gates Foundation has also funded us at RenBio because they see us as a low-cost alternative to get antibody and protein therapeutics to people. DARPA likes it because it's a fast way to get them. Because as we saw now with COVID-19, the nucleic acid like RNA can get to people much faster than proteins. They're programmable therapeutics. And so they saw the potential of what DNA could be if there's a new infectious disease outbreak, a new virus. A DNA version of an antibody or protein would get into humans quicker, whether it's for the general public or for military use, because it's still DARPA, which is part of the Department of Defense. They can roll that out much quicker, and it could be long lasting. You're not thinking again about infusions at like a battlefield or something like that. You get an administration at the base. So they came in early on and really validated what we were doing, which was huge for us.
Yeah, no, that's huge. We had two questions from the audience. One, which is, how do you know how long lasting it's going to be, and kind of why isn't it permanent? Like what, is it, do you have the ability to tune it? Like, do you know kind of person to person how long things are going to last? And then just as a simple question, what does the Ren in RenBio mean?
Yeah, those are great questions. So how long lasting it is. So that's evolved over time. We just gave it a shot to see what would happen. And the first experiment, it was about 3 months in mice. And back then antibodies were about 3 weeks. So we said, okay, that's a good start. But when we started RenBio, we said, we really need to innovate on this and make it more long lasting. And so our team is led, our CSO is Rachel Liberatore, and she's done a fantastic job leading the scientific team here. And we're able now to extend that duration for antibodies up to a year after a single administration in animals. So it's a balance of a lot of work on the DNA vector, really thinking about our target bioreactor, if you will, is the muscle cell. So making the muscle cell very conducive for expression. We did rely on some data that was out there. So DNA vaccines had been in humans before. We're not doing vaccines. We're doing more of this approach where it's rather than have the immune system recognize what you're giving, you actually want it to be like a Trojan horse that's silent and quietly pump out. But we had a lot of data knowing that circular DNA did not integrate because of these vaccines. So we were basically taking concepts that existed, but combining them in a new way. So there are elements that were de-risked, but the combination was new. So we knew it wasn't going to integrate, but that's going to be part of the toxicology studies we'll do. But there's a rich data set about circular DNA to show that should be okay.
Yeah, it wasn't like people had done what you had done before, but they have done kind of pieces of what you've done. And you can kind of make the extrapolation of like, well, if they do this over here, this should be what this looks like. And this should be what this looks like over here.
And that goes beyond DNA. So it's for the DNA part. And it's also in the beginning what you deliver, right? If we were thinking about things that had been in people before, the biggest place where molecules can die is in that first phase 1 study. You don't always know if your animal studies will translate. So we were taking things that we knew had a great safety profile in humans and also a DNA concept, but now combining them in a new way. So we had elements that were de-risked, but we're innovating on how we put them together.
Yeah. Well,
Um,
I will say as a tip of the hat, so I know Stephanie Tompkins, who's the director at DARPA, actually likes to tout RenBio as one of their success stories for their biological technologies office. It was in her remarks to Congress last year. And I was at a DARPA conference about a month ago or whatever, and they sing your praises in terms of technology development. So that's a very nice endorsement. So they tend to like folks that deliver, and they tend to think you guys are one of those. So that's a nice
that's
one
awesome.
right there.
Thank
Yeah,
you
that's
for
pretty
that.
great.
That's a good.
It's not me. You
Warm
can
fuzzy.
you can thank Dr. Tompkins directly, I suppose.
Yeah.
So okay, so
Should I want to get
right?
to this other question as well about the
Oh, go
Ren.
for it.
You want me to do that one
Yeah,
first, or
yeah, go for it. Yeah, let's do that one.
Yes.
because that was fun too, right? Because all of a sudden we're logo designers at the same time
Yes.
and thinking about company names.
Exactly.
Adding something new to your portfolio.
That's a really fun time.
Those
We're
are
not
the only
artists.
people in biotech that won't tell you that it's the hardest part of the industry.
It's, we thankfully found some designers to help, but so Ren actually comes from Confucianism, and basically in a very literal sense it means person, like man, and there's a symbol that's actually in our logo that looks like the symbol for Ren from Chinese.
Okay.
And if you have two of them together, it starts to mean something different. It starts to mean humanity and mankind. And Ren has a lot of different feelings and meanings. Like the translation is very difficult. It's humankind, it's benevolence. It's the best feeling I can have is that altruism that a mother has for her child or a parent has for her child, that unconditional love. And so RenBio was basically a marriage of that altruism, benefiting mankind, doing something for humanity through biology as well. And so that's the Ren. So some folks know that very well if they come from an East Asian kind of background or know the languages there. So it's a little tip to that as well, and where our co-founders have come from.
Well, that's genuinely thoughtful and pretty cool. I like that. So in the middle of all of this, we basically get hit with a pandemic, right? So what was happening inside the company when the pandemic hit? Because you've got this cool Series A story that's kind of just like enveloped in everything that's going on with the pandemic. So I really want to hear how all of that unfolded.
Right. So pandemic in March 2020. So end of 2019, January 2020, we were getting ready for a Series A raise.
Yep.
So we had our seed, we had additional funding from DARPA and the Gates Foundation that really helped prove out our technology in animals. And we wanted to take it forward and start working on the clinical development plan. So a lot of 2019 was deciding, what the lead indications would be to prove out our platform, how we want to take this forward. And we were thinking a lot about the different commercial indications, infectious diseases. We always came back to because we just have a background in that from our co-founders and our CSO and myself in virology. And we were developing the plan and about to hit the road. And then we all went home. We didn't stay home for long, but we went home for the pandemic. And it was a little, and I was in a panel recently where people were talking about, we all thought this was going to be 2 weeks and we'd come back to the office in a couple of weeks. This is where the cynicism came out for us because we were virologists. And we said, I don't think this is going to be just a few weeks. So if you follow the news, David Ho in Columbia, they have done phenomenal work in the SARS-CoV-2 COVID space, and it really laid a lot of the groundwork for understanding the mechanism of action, developing antibodies against the different variants, discovering the new variants. They've been kind of at the forefront of this work. And so we had to make some decisions. At that time, I think we, like many people, said, what are we going to do to help stop this pandemic? What can we do? And our investors at the time, like I said, they had a philanthropic bend. They wanted to do something as well. So as we started thinking about, okay, we want to raise to bring our platform. So we call it the myotechnology. And it has two names for it. That is two reasons. One is it's make your own, because we want to empower individuals to make their own antibody therapeutics, but it's also play on myocytes because those are our in vivo bioreactors. So something for us as scientists, but something hopefully commercially. Someone later down the road, I'm sure, will have to spend a lot of money and they'll either tell us it's great or they'll change it again. We'll see.
That's fine.
Yeah. So we just made a team decision as a team with the investors. We said, look, we want to do something for this pandemic. Let's bring an antibody forward. We knew vaccines were going to come out. They're going to be great. But from our understanding of the field, we knew there were a lot of people that were already infected that were going to need treatment. We also
Yeah.
knew there were a lot of people who are immunocompromised, 8 million people in the US alone who are perhaps the most vulnerable, who won't
Mhm.
benefit from those vaccines. And they need something almost as a preventative agent, and antibodies can serve as those as well if they work.
Yes.
Yeah.
So we developed a two-pronged approach. We in-licensed SARS-CoV-2 antibody from Columbia University, from our co-founders. And we said, look, ultimately, we want to develop this with our mild technology platform, but that's not ready for humans today, and we don't have time. So as a stopgap measure, let's take this antibody forward into humans in its current form while developing the next generation approach, kind of modeling what we were thinking at Iron Diamond in the early days for HIV, but now doing it for SARS-CoV-2.
Right.
And the investors are on board. We brought a few new ones involved. Again, it was a mix of an interesting group of high net worth individuals, some companies that came in as well. And we closed a $24 million Series A in December of 2020 to take forward a SARS-CoV-2 antibody and take forward our Myo platform for that COVID antibody.
Right. So what was that discussion like? So you've got a dichotomy there that tends to be a hard discussion, right? You know, investors like to fund one thing with one plan that's going to go in one direction. And if they say, this is what we're putting our money on, we want it to be on the one thing, don't give us two things. So how did that conversation go? I mean, it sounds like you had a very measured approach. You had pretty open discussions. And if I remember back to those days, the fate of the vaccines was very uncertain. And there was a level of uncertainty around, we don't know what we're gonna need, right? I think the initial guess on vaccine efficacy was somewhere between like 50 and 65%, right? Which would have put us in a much different treatment paradigm than we're in now, right? You know, nobody expected the 95%, which is great. But so what were those conversations like, really upfront and as things progressed, right? Because as you said, I don't believe you've said that the antibody isn't in development anymore. So how did that roll out?
Yeah, so many internal discussions with our leadership team and with the investors. We just kept it very open with them and talked back and forth because it started to turn into a partnership, right? We had to figure this out, and it became kind of a maybe encapsulating the Ren in our name, right? Like, what are we going to do for society here, right? What is our ultimate mission and goal here? Our thought going into this, as you said, the vaccines, no one knew if they were going to work. There were some antibodies coming out, right? Regeneron led the way, Eli Lilly as well. A lot from our HIV background, very early on in 2020, we said there's going to be resistance and there's going to be variants, because the one thing we always said at Iron Diamond is the virus will always find a way. No matter what you throw at it, the virus will always find a way around it, and you have to find a way to deal with that. And SARS-CoV-2 was different. In the early days, we said, look, it doesn't have as high as a mutation rate. It shouldn't be an issue, but just the sheer enormity of how widespread the infection was, that there were just multiple vessels to replicate in and create these mutations. So we went in early thinking, maybe we should do a bispecific antibody so we can hit it at two different places. Or are there very, very broad antibodies? So we went in saying, all right, get the broadest antibody possible, look at combinations of antibody. Everyone's developing individual antibodies or hitting a specific target. If we're going to make any impact at all, where can we come in that's differentiated and different? So we're looking at bispecifics and hitting different targets. And that's what we did. And that's what we took forward. Soon as the variants started coming out one by one, things started dropping, right? The vaccines were good, but the antibodies were dropping. The alpha, beta variants were coming out. Some of the early Eli Lilly antibodies took a hit to those. Delta was the big one in the summer of 2021. And a lot of antibodies dropped at that point, but ours withstood it, which was fantastic. And that's what the design was, right? That's what we wanted it to do, is to withstand the variants. And so we charged forward. And then Omicron came out, which still makes me shudder because that changed the game for everyone. If you watched, Omicron pretty much knocked out all the antibodies. It was honestly a dark time in the field. I think as a field, we were all like, what is going to happen next? The virus has found a way. And now with the subvariants of Omicron, even more of the antibodies are getting knocked out. And so when Omicron came, our lead antibody took a hit in its activity. It was still active, but it changed the benefits. In the early days, it was super, super potent. We were looking at sub-Q formulations, not wanting to do IV, things like that. When it started to reduce by that much, we said, okay, now we might have to give a higher dose. We have to look at IV formulations. Is this going to happen again? And it goes to what you were saying, Derek, right? We had two programs we were developing. And one thing that really emerged is the Mayo platform was variant proof, right? It was variant independent. We weren't there delivering that was a platform that can deliver whatever the best antibody is. It's not going to succumb to any one antibody because you could rapidly plug and play a new antibody whenever you want it.
Right.
And we realized just the resources it was taking, we didn't bring it to the clinic. We stopped before that. We started the CMC work and we said, look, you know, one of those questions of when you have to decide if a program's going to die, it was a really tough decision that did not come easily. And we said, look, we've got to just cut our losses and then focus on the platform. That for us is where the real value is going to be in the long term, not only for COVID, because you can plug in different antibodies that work with whatever the best variant is, but also for future infectious diseases and outbreaks, and also for any other disease. And so it was a lot of those variants that really caused us to say, there are other groups that are better capitalized that are going to do this, right? You had the big pharma players there, and the market in a day could change, right? It could be huge or it could be little, depending on the variant. And as a company like ours, that's not going to be a great place to raise money.
And also the further you go, the harder it is to say no, right? That the further you went into development, the harder, the bigger climb it was going to be to say, this isn't going to be there, right? Because you have, you know, ultimately you would have had, you know, dwindling, you would have had less cash to do something else with. You would have been closer to any sort of milestone that you could talk about, right? You would be, you know, how do we get out of the no man's land of something? And, you know, the kind of trick of being able to raise money, you know, how do you answer, you know, these kind of investor questions? The further you went, the harder that was going to be.
Yeah, I think you hit the nail on the head. And, you know, this isn't a new phrase to us. People have said this before, but if you're going to kill it fast. And that's great to say from the outside. I used to say that in the early days, but when you live it, it's very different because you're thinking about the investment in the money, your time, your team's time, right? And everyone has dropped everything and is dedicating a lot. And, you know, ultimately, for the benefit of the company overall, you just have to make those types of decisions. And
you also, you know,
sorry, I was just
no, go ahead, Jennifer.
Thinking about the potential patients, right? And like in some aspects, there is a like, will those patients be treated by someone else stepping up, right? And that, I know that has to be part of your, you know, your calculation. And there goes back to your balance, right? Of
exactly,
science and finance and
and where are we going to have the most impact, right? Where we make the most difference for humans,
right? Okay, so where are you now, right? So you're in the middle of you've got your Series A. You know, unfortunately, we have stretched outside the realm of, you know, the layup IPO or the $100 million Series B that you cash, you know, 2 weeks after you raise your Series A, right? That is not what the market looks like right now. So what are you looking at now? You know, and what are you kind of thinking of for, you know, for your next steps? So what do you have kind of cooking, and what's your kind of outlook for the next little bit?
Yeah, so during the Series A, even with all of the work that was going on with the COVID antibody, we kept developing our platform. So what that allowed us to do is basically lock in our clinical tech or clinical facing tech. So we locked in our final vector designs. We locked in our delivery system as well. We filed new IP on that and pretty much built that up. And then did kind of started thinking about clinical indications. I should say just a shout out as well. We very luckily were part of the EFL program, the Endless Frontier Labs through NYU, led by Deepak Hegde. It was fantastic for us. It really gave us access to an incredible wealth of knowledge and investors out there and mentors that really helped us hone in on our story and our technology and where it could have that impact. And so a lot of the Series A was spent doing that through 2020, 2021. And now we're thinking about bringing this to the clinic. So we're starting to raise a Series B to do just that. Not the climate to raise a Series B, but there's money for good science. And we're seeing that good
I think
science
so.
is getting
I think I think the money will follow things that are good. And I think Series B now are actually genuinely looking for good science and good data. It's not really about the FOMO. The levels of FOMO aren't necessarily there, but I think when you ratchet down, you actually look for things that fit kind of a profile like yours, right? And I will also say that everyone that I've met that has gone through the Endless Frontier Labs has wonderful things to say about it. Like 100% of the people that I've met that have gone through that program love it.
Highly recommend it. And we're pretty practical. For being optimistic, it's practical still. Like we weren't looking to do this mega raise for no reason, right? We're like, what is going to get us to the next value inflection point? What would be meaningful for us? And what would be the next way to de-risk this technology? And what do we do to get there? So we've opened up a round to raise a Series B, but luckily for us at the same time, because of the market, we relied on some of our skills and experience from my nonprofit background, where we also just applied for a lot of non-dilutive funding. And in the last year, we've gotten several $1 million, again, from DARPA and the newly formed New York State Biodefense Commercialization Fund, because they see the impact of what we can do. So that's really for us, changed the game. It's just extended time to really raise a nice Series B, do it the right way, not feel like we're pressured. Because we have this non-dilutive funding to carry us through. So it's still thinking about that balance and kind of leveraging different investment vehicles when you can to ride these markets.
Right. Flexibility, creativity, and in your case, the ability as you go out for your Series B, you'll be able to answer questions better, right? And you'll have more complete answers, hopefully, for those investors.
Absolutely.
Yeah. Right. So, okay. So here's a hard question, right? So you're a platform biotech company, and now is the point where, as you start to go into the clinic with things, these now get to be, bigger bets, right? So how do you think about being a platform company when you're about to be clinical? Do you think about things within a lead indication? Do you think about advancing multiple indications? How do you think about that strategically? And then how do you tell that story?
It's something we talk about a lot. Like me and the CSO, Rachel, we have had debates on this, and a lot of what we believe ourselves, because we need to be authentic for what we think is the right thing to do, and then conveying that, like you said, to investors. And it's been different depending on who we talk to. For us, we see this as having a huge impact in multiple disease indications. So what we're building is something that's long lasting. We're not looking for just one indication and to be done. Right. We think this can impact a lot of different diseases, and we're building a platform for that. So we're actually taking in different stages, 3 different indications that are of high value interest to us to prove out the platform. The first one, and it's interesting when we'll meet with VCs, of course, there's questions on market size and commercialization. Some of them huge commercial markets, other really small commercial markets. But the value they'll bring is we have high reason to believe it's going to work in that first clinical study. And that's of tremendous value, not just for that disease, but for the platform, right? If you show it works in that first indication, that opens up many other possibilities. So we're taking the multiple indication route, but different investors we'll speak with really want to hear what your first one is.
Of course.
And they want to base everything on that, which is also another model to do, which isn't wrong in its own way. And so we talk about that, but I just see us as a much bigger thing here. This first one is just the first step to creating multiple products. That's the goal for us here.
Well, I think that's really smart in a lot of ways, because first and foremost, you're looking at something that can fundamentally change how things are treated. A lot of times in biotech, you think about the top line numbers, efficacy, safety, that kind of thing, and you don't really get into the understanding what an infusion means once a month. Right. It means or every 2 weeks, right? It means every 2 weeks, somebody has to actually go someplace, sit down and get an infusion, which is really non-trivial on the part of patients. So I think if you think about it through the lens of patient experience, patient care, et cetera, there's a lot of big things that can happen here in terms of, how a patient lives their life with a particular disease, holding safety and efficacy constant if we can. Right. So there's a lot of innovation there that I think is really smart about, if this works for this kind of disease or this kind of treatment, what does that mean? Does it open a window to these other indications that are very similar?
I think it's really interesting because, as scientists, you say, well, this drug works, so people will take it, right? And that's just not how it works. From the HIV world, we're down now from, if you remember the 90s, where people are taking 10, 20 pills a day, carrying around coolers, having to eat it, with food. There's timers going down to a once a day pill now for a triple combination therapy. And still adherence is an issue. And resistance forms. And there's market research on that. And the reason that is a lot of different reasons. But one of them is people just don't want to be reminded of being sick. And every day they take that pill, they're reminded that they have HIV. Some folks are okay, some aren't. And people will take drug holidays. They'll take a vacation and they won't take their medicine because they just don't want to think about it. And we don't always think about it. I don't always think about that experience as a scientist. I say, wait, it's a medicine, it should work. But it's the same for
Right.
antibodies. Like
Yeah.
if we can change people's existence, and antibodies is one thing we're working on, but there's actually a lot of protein therapeutics with really short half-lives that people are taking daily injections, right, in their abdomen.
Yes.
If you've
Yeah.
got someone, we're not looking at diabetes, but if you know, you might be
yeah.
at a dinner with friends
Yeah.
and someone has to casually step away and give themselves an injection. No, like that is not a part of life. That's something
Right.
we want. And
It works 10% better. All you need to do is take your pants off, stab yourself, do this, and then sit, and everything will be fine. Right. You know, and
right.
it's like, well, it's better. Why won't people do that?
Where are you going to do it? Or, you know, you might be on vacation and you're in an amusement park and where, like, right, how are you going to take your pants off to get like an injection in your
Yes.
quad or your stomach? So we're
Although
thinking
having been to Six Flags this summer, like that might not be a concern for some people.
Yeah.
Okay, fair. But that's the thing, right? So if you can
Yeah.
get one injection, be good for a year, that just changes your outlook on life, I would say, right? And
Yeah.
that can work across many diseases where, again, going back to where RimBio started, people are innovating and doing incredible things to address unmet needs and indications that don't have medicines and things like that. But just living a normal, healthy human life is something that still needs a lot of innovation to help, right?
Mhm.
Both in the developed world and for the rest of the world as well. And that's something we want to do.
Well, even
Yeah.
we've had conversations on this program about, you know, like when you talk about the developing world, like those are problems. But even to a lesser extent, if you live in Staten Island and you need to get to Columbia for an infusion, right? I mean, like these are real barriers, and
Yeah.
they're not trivial for the people that need those treatments.
Absolutely. And if you're working, like you're taking a day off every single time.
Right.
Like it's a 30-minute infusion, but if you're coming from New Jersey to New York to go to Mount Sinai, right, or MSK, you're sitting in 2 hours of traffic, you're getting prep. It's great because it works, right?
Right.
But we shouldn't stop there. We can't.
Yeah. No, I think, you know, we talk about patient experience on the show over and over again. And, you know, it is such a driving force for innovation. And thankfully now it's getting the airtime that it should. Right. And
So
I think that's a very, very good thing. Okay, so talk to us a little bit about, you know, we've gone through this with this whole thing, which is great. So talk to us a little bit about how the heck you did all of this in New York at a time where, you know, J Labs and Launch Labs and none of those things existed, right? So how has it been to kind of put all of this together in New York? And, you know, it's okay if you kind of, you know, scoff a little bit now at the fact that these young whippersnappers have all these incubators available to them when you didn't have it when you were starting.
oh man, incubators would have been great. Yeah, there really wasn't much at that point. I didn't realize, I was like, you know, I'm like, oh, this is a founder-led biotech. That's essentially what it is and
Right.
what people are doing, because we didn't have the
You did that
benefit of
before it was cool.
yeah, we didn't have these institutional VCs that said, this is how you set it up. This is what you do. You know, one, it's just a theme that we've always done, coming from basic research and not always having the answers and learning how to figure it out, doing the drug development, doing the company formation. It's a style that we've gotten comfortable with, which is not a scientific approach always, right? You like to have things neatly done, but it's an innovative and creation side of it, which I really like. And so you just figure it out. Iron Diamond was a great place because that was a freestanding nonprofit institute, even though I had an affiliation with Rockefeller, now Columbia. So I did have mentors and resources there about how do you set up payroll, right? And like insurance policies and things like that. So I had mentors and I was starting to translate what that meant for the commercial side, because that was very different. So you just rely on your network and you build your network like crazy, right? Just starting with your friends. And you go from there. New York City has been great. I really liked it for a few reasons. The New York biotech ecosystem itself feels like a startup, and I like that, right? I like that it's still in that new formation place. You can see it. It has legs now. It's really starting to grow. And I really love being on the ground floor of stuff. I love coming in when something just starts. I really like kind of seeing things get born and watching them come to realization. And I feel like that's what New York City has been for me. There have been a few things that have really helped me. We were part of the Alexandria Center for Life Sciences in Manhattan in the early days. There was someone who's moved now to the North Carolina site, Blake Stevens, who was there running Launch Labs when they formed. He brought me
he's
in.
great.
I love him. He was great. He said, Neal, look, all of our launch lab companies, they want to be where you are now because we were the next stage over. Why don't you come
Yeah.
speak to them? You know, and I got to meet that network. He created this New York City Founders Network where you can meet other people in the New York ecosystem, which has really kind of connected us in a great way. And I think it's been very interesting to hire here, right? You get a lot of people from the universities and academics who want to come.
Yeah.
And New York just brings out a special type of personality. Like it is a driven group here, an excited, fun group that has, you know, love for science, but a love kind of for all things. Like we have someone who's an like competitive rugby player, someone who's a professional gamer, someone who's a DJ on the side. Like you get the gamut here in New York City, and that's something that I don't think you get everywhere that I really love.
Yeah. It makes at a minimum, it makes the office more interesting, right?
That's true.
Did you have trouble, or was it still as easy? Because you obviously have grown and expanded throughout sort of COVID and, you know, remote and in the office. And I know you have a lot of scientists on your staff. So how did you all navigate that process?
You mean growing during the pandemic or? Yep. Yeah, we continued to hire through it. I think we were home for a month or two, and then the scientists came back because we were working on COVID. Like we had to be there. So we needed to hire people, and people were looking for jobs back then. People were still graduating from grad school. People were still looking for new jobs. So like everyone, all of a sudden, everything turned virtual for the interviews, but they were willing to come on site. It was harder for people that were in other states. Even in those early days, if you remember, like every state had different policies.
Yes.
Mm-hmm.
And you couldn't always move to a high-risk state. You know, from New York, you had to quarantine at one point in the very early days. So that was something that we were trying to deal with, but we didn't have any problem hiring during the pandemic at all. I think scientists were looking for something new. And honestly, scientists wanted to work in this disease.
Yeah.
So as we only have like 2 minutes left, Neal. So as we look forward, right? As you look forward to the next, I'm like, how many years out do I give you? I'll give you 5 years for RenBio.
Oh, wow.
Yes. And the New York, and sort of your place in the New York ecosystem. What do you see on the horizon?
For RenBio, I think by that point, we'll have multiple products in the clinic. That's the goal. We'll have the first one in about 1 to 2 years. I'd like to see the second one in by then. And I think for us, that's going to be like most products. That's a huge place to de-risk the technology and can really kind of expand a few things. One, it'll reveal new indications to us when we start seeing what the human PK data looks like. I think it's also going to expand partnerships, right, from biopharma partners that have antibodies and proteins of interest or need a new way to deliver them. So I'm really excited to see this get to the clinic because I think that's going to just reveal a lot of information for us. And I'd love to keep on growing here. We moved to this space with the intention to grow even bigger. We're in Long Island City now. And that's the goal, that we're going to do that here.
You did that before it was cool too.
I did. We were one of the first times. Opentrons was here, which was doing great work for COVID, but we were definitely here when construction was still happening. But it's like the dream. It's like when you move to the up and coming neighborhood as a resident in New York City before it gets too popular.
Yeah. Yes. Well, yeah, no, go ahead.
You had a second question. I was trying to remember what it was, or was that?
Sort of the growth of the ecosystem.
Oh, yeah.
You said we seem early stage. So tell us what you would like to see NewYorkBio or the ecosystem do over the next 5 years.
I would love to see in 5 years. So we're seeing IPOs already. I want to see a few more of them. I want to see some big acquisitions that happen as well. Yeah. And I want to see the next generation of companies. I want to see all those folks who learned and earned all that experience in this first generation of companies start to form their next set of companies. Yeah. I think that's going to really be what then kind of seeds and really nurtures and grows this community. And I'm hopeful that in the next 5 years, we'll start to see that.
Yeah, we've talked about that a lot too. And I liked as Derek knows, my favorite term is the flywheel of C-suite talent, right? To get moving and turning so that we're doing it again and again.
Yeah, absolutely.
Well, we are at NewYorkBio, Derek and I especially are appreciative of what you all have done for our community and how engaged you are, and especially your conversation today. We hope everyone has a good week. We will see you next week. And thanks again so much for joining us. Thanks, Neal.
Thanks, Neal.